So I will be talking about non-invasive solutions to brain-computer interfaces. Okay, I'll scroll through to the first title page. Okay, so the title is non-invasive and minimally invasive neurointerfaces for neurorehabilitation control. So they developed together with this team and funded by the mega grant obtained by Mikhail Lebedev. It's a special way to prominent scientist in Russia. For this funds, we have created a Center for Bioelectric Interfaces. It's all part of the High School of Economics in Moscow, and this is the work done together with Ossadtchi. Okay. So, well, because it's neurointerfaces, you probably guess it has something to do with the brain. So the brain, you can see your brain. If you look at your MRI, you can see the brain structure. The brain contains many interesting different structures, but that will be concerned mainly with the cortex, which is outlined here by the red dashed line, which is a thin gray layer on the very top of the brain. Okay, the area of the cortex is approximately 2.2 squared centimeters, and the volume is equal to a big jar of beer, like 5.5 liters, the area of the brain. Okay, so the brain, the cortex comprises about 100 billion of neurons, and it also has at least 10 as many astrocytes and other glial cells. And so this is a hugely complicated structure. Some people say this is the most complicated structure that exists in the universe. And given that there are a lot of brains that exist in the universe, so I don't know how we can exactly reconcile this, but anyway, so this is a complex structure, and it's not only the structure, but it also has a function, and the function here, for example, is depicted codes. So, and the function is produced by the neurons which activated and generate in so-called action potentials. And then when these neurons communicate between each other, they create basically us, okay? But when the brains were created, nobody actually thought about the need to measure their activity. But it turns out that we can actually see it. We can see it, we can visualize it, we can use it for various purposes. In order to understand how, I'll show you this example here, which is from Buzhaki's book called Streams of the Brain. So now if you imagine that there is an audience and you're clapping in response to some performance, then the clapping first of all tends to synchronize. And then if you average each individual claps, you know, within some time beams, then you will be able to see this kind of curve, which has an oscillatory nature. It comes and goes, comes and goes, comes and goes. How fast it goes, how fast it changes, it's basically the frequency of this oscillation. This curve is changing at 10 Hertz because one period happens within approximately 100 milliseconds, okay? And if the brain activity, the activity of the neurons is synchronized this way, the same as clapping, it could be heard far away, okay? Well, this is a very, very, of course, loose analogy, but so because I don't know the educational background of the public that joined this seminar, okay? So now, once you have this neuronal assembly that is acting synchronously, then it produces those action potentials that are forming so-called local field potentials, which are exiting the head and causing the currents, okay, the exit in the brain, the head, making it through the skull and forming the potentials right on the skull. And so then using the electrodes, we can measure this potential difference, okay? These potentials are extremely weak and specific equipment is needed for that. In order to understand why they're weak, you can look at the parallel phenomenon, which is also electromagnetic phenomenon, but at different frequencies, that if you take a mirror and the light, most of the light will be reflected from the mirror that falls into it, but some amount of light will pass through. That's pretty much some of the current that is reflected back into the brain, back into the inner part of the head from the skull that is non-conductive or poorly conductive as compared to other structures. It gets reflected inside, but some of it gets outside and produces this difference of potentials, okay? So the ways to register in electroencephalogram. This is how it's called. These attempts been made long ago, starting from the second half of the 19th century. And Hans Berger, in the beginning of the 20th century, recorded first human encephalogram invasively and registered this rhythm that he called alpha rhythm because it was, I guess, the first rhythm that was found. That's why it's alpha. Then several other rhythms were detected like theta, beta, and others. They have all different frequencies, but the name, I believe, alpha is due to historical reasons. Okay. The modern devices, the modern EEG devices, you can see here. So they're very small. You can even fly your own airplane and have your EEG with little of interference. And there is high-end devices which allow you to record the activity with 128, even 256 electrodes from the skull, and those are also pretty compact and very reliable. Okay, so the interfaces that we will be talking about today, that's the EEG, right? The EEG is something, the technology that allows you to measure brain activity, and measuring brain activity, you can use it for different purposes. In particular, you can use it to create neurointerfaces, okay? Neurointerfaces are devices that you can use, systems basically, that can use the external devices the thought. You know, you can think, for example, about your right hand, and if you design your algorithm properly and connect your electrodes properly, then the system will recognize that you're now thinking about your right hand and will make some action. Okay, so how it happens and how it becomes possible. All right, so you saw those rhythms, right? I showed you. So the neurons tend to get activated and tend to get synchronized to produce these rhythms. So the brain is organized in such a way that there are specific regions that are responsible for specific functions. Okay, it's a very long ongoing debate about the actual specialization of individual region and between regions. So the truth is in between. Indeed, there are some well isolated representations in the brain of different body parts. Okay, and that is one of those pictures called homunculus. Are seeing the sensory motor cortex. See how different parts of the homunculus have different representation of different organs. Okay, so now imagine that you're thinking about your right hand, for instance. Okay, then in the left part of your brain, sensory motor cortex, you will see what we call the desynchronization of the sensory motor rhythm. Desynchronization means that the amplitude of rhythm will drop. Okay, something's going on with high activity, and then once you started thinking about your right hand, it will drop. We will know that you're thinking about this drop right above the brain area that is responsible, where your right hand is represented. Okay, well, of course, this is a very idealized picture. Things, in fact, are significantly more complicated, but the basic principle is this. So registering the reorganization of the structure of brain rhythms, you can detect certain states of the brain that are associated, that you can associate with specific commands, and then you can fulfill these commands. Okay, so just to see how we can visualize brain activity. If you take EEG electrode and then solve the inverse problem, then we can see something like this. This is actually the activity during motor imagery in beta band, in the band from 16 to 25 hertz. You can see how different brain areas highlight, and you can use this information to decipher brain activity and to make certain actions, okay, to have your external devices perform certain actions. You can see very interesting different patterns like wave-like patterns or like swinging patterns and stuff like that. All these can be recognized with modern mathematical methods, and you know, it can be turned into the interface in control commands. Okay, so let me switch to the next slide. Yeah, another well, you can track the rhythm, and you can have a person who, I will launch this now. You can even play games. I will scroll so that you can collect the bitcoins here. Like Xenia is sitting, and in another game, it's a doodle jump. You can control the ball moving to the right or to the left, and you have to do so that it goes and reflects from the platform up, and you score here. So you can do it with just the strength of your thought. Okay, it doesn't always work, of course, because it's a complicated thing, but in general, you can acquire the score and it goes up, whereas if you leave it without control, it goes big negative. Okay, so I'll stop this and go to the next slide. Okay, so these neurointerfaces are useful in several applications, but the most, I think, the most interesting and most socially important application is the use in rehabilitation of patients after stroke. Okay, the stroke is something, this is I've taken from Recovery After Stroke, 5 Tips for Rehabilitation article in the Frontiers, very nice resource, Frontiers for Kids. I enjoyed reading it. So the main idea that at some point the artery, for example, becomes clogged and the brain doesn't receive the oxygen and is not capable of flushing the residuals of its activity. Okay, so and then this area of the brain basically dies. Okay, or in other cases you may have a hemorrhage that is on the one hand presses on the brain tissue, and this also causes destruction of the brain tissue and loss of a function. Okay, so very often the stroke results into the disability of people, in a sense that the people lose the ability to move their hands, for instance. And usually it's lateralized, usually it's right hand or left hand. Sometimes it happens with legs as well. Okay, so the idea for rehabilitation is to basically recreate neural circuits that were damaged by the stroke, recreate them so they work exactly or similar to the way they used to work, and you're able to recruit all necessary pathway to make your hands move again or feet move again. Okay, and so there is this product by RecoverX, for example, from GTech, that the person is wearing an EEG device and thinking about his or her left or right hands, and the avatar is moving, giving her reward, positive feedback if she thought about this properly. Okay, so when she thinks about it, the sensorimotor cortex gets activated, and this activation increases the blood flow and blah blah blah, and there is a legend which is now getting the support from the literature that this kind of exercise, this motor imagery exercise, or in case of patients, it's not really an imagery, they're just trying to actually do the movement, but just can't. So this motor imagery improves the rehabilitation process. Okay, there has been a study where an exoskeleton for the hands was used. So here we have a exoskeleton that sends you feedback also, because when you are thinking about your right hand, for instance, it's activating your right, a grip of your right, okay? And then the proprioceptive information is traveling back to the brain right in the moment when you decided to do the movement, okay? And when these two information flows meet in the brain, this causes extra plasticity that is utilized to restore the brain function, okay? That's for the right, this is for the upper limbs, okay? And for the upper limbs, this is more or less done, although there are clinical trials now that are trying to investigate the efficacy of brain-computer interfaces in this task, okay? We have also, if you talk about right hands or left hands, we have developed this was thought to be a toy, but in fact, this is an interesting application for the post-stroke patients who can play this game. This is so-called a neuro wrestler. So I'll show you the idea. You can like one person, the two people, those are the healthy ones, but sorry, just a second, because there's sound, I want to get rid of it somehow. But anyway, so they're thinking about, Nikolai is thinking about his right hand, and Dmitry is thinking about his right hand as well. And they're trying to incline the bottle into the direction of right, basically. And if they think properly, if the brain cortex get activated or deactivated, the neurorhythm gets deactivated properly, then the bottle will return, will bend onto the side, so that the proper person will win. Okay, the properly thinking person will win. Okay, so and this can be like an exercise, which is instead of cards or other kind of games played by the post-stroke patients in the lobby of a hospital. Now Dmitry wins. Okay, so but and this is actually on the one hand this is a game. On the other hand, this is a this is potentially an interesting solution as a neurorehab training for the patients with for upper extremity movement deficit after stroke. Okay, the with we have the interface which controls the exoskeleton, and that because exoskeleton is of low limbs, this involves motor imagery of the legs or feet, and this is foot and leg representation is lower, and it's in between the two hemispheres. You know, it's in the inner wall of each hemisphere, and it's further away from the sensors. And if you think about how the person in exoskeleton, the person stands on the skeleton, a lot of muscles get activated, but we will return to it later. I'll just show you the how it in general works. So you are looking for activity that's the location of the topography, but it's basically the mark location of the electron, but the signal of interest spread. It uses the spectrogram of activity during the rest or during the motor imagery. As you can see, during the motor imagery of feet motor imagery, legs motor imagery, the mu rhythm, the alpha component, the 10 Hz component, and beta component of the mu rhythm, they desynchronize, they disappear. Okay, and based on this change of the structure, you can control any external device, in particular exoskeleton. Like here, we participated in the Neurothlons twice with this interface with true patients. So this Artyom, he is a paraplegic, so he can't move his feet at all, his legs at all. And so he was able to control this U-boat to make it around the bushes, underwater bushes, because when the boat crosses and gets onto the bush, it slows down. And there was a race, and he was number 2 in this race. Well, I mean, I will not tell you the reasons why he was number 2 and not number 1, but I can think that he was number 1. Okay, so then there was another game next year that there was a like set of spaceships that were flying through a asteroid band, and depending on the type of asteroids, you have to be in a certain state in order to pass through it fast. Okay, so that was in fact 4-state BCI. Okay, where you can control exclusively your brain. Okay, and he will and Artyom was also number 2 with our BCI. Okay, so I think the performance of the BCI that we have developed is pretty reliable. And here Nikolai Smetanin is the key developer of the software for the BCI. Okay, so all right, so leg BCI, the problems with this is that if the person is standing and lesions get stressed, a lot of artifacts in your EEG that you have to get rid of. Okay, at the same time, when the exoskeleton, it's very powerful actuators that actually move the legs. Okay, when they kick in, you have a lot of artifacts. Okay, also, so we had to get rid of those artifacts. We have to figure out how to do it online, and you know, many technical problems that we had to resolve. And finally, we came up with this kind of a system that basically is based on the smart BCI EEG device. That is this very compact EEG recorder that records 32 channels, but you can actually do with much smaller number of channels, but we are using this one for now. And we are recording this, we are extracting the sensorimotor rhythms, we are making a decision based on this, and we are powering the exoskeleton and telling the exoskeleton to make a step and not to make a step. Okay, and so then when we bring together the desire of the athlete or of the patient to make the exoskeleton, to make the move, and the actual movement exoskeleton that sends back to the brain the proprioceptive signals, okay, then we believe that, and the studies show that this kind of a solution should improve the neurorehab process, okay? Then even if you link this with what Kate has described about this system for registering different objective parameters, including electromyography, you can also study a lot of interesting quantities such as corticomuscular coherence, for example, and see if it can be an indicator of the progress of rehabilitation progress. Okay, and we are in the process of developing this technology and introducing all this setup into the line of exoathlete products right now. So we have tested our solution. So we put the patient in this exoskeleton. We ask the patient, sometimes we ask the patient to go, sometimes we ask the patient to stop, and the patient has to do both, you know, so that we want to test, so we could test the specificity and sensitivity of the device, okay? It turned out it works pretty well. I'll show you the ROC curves, but to show you how it works, you know, how how it actually works here. So here the person was instructed to stop, and he stopped. Okay, now the person is instructed to go. Now he's successfully thought about his feet, or actually tried to move his feet, because he doesn't have to think and limit his movement, because even if he tries to move, they don't move. But now they move. And we tried it in 3 patients so far, and all of them were successful. Well, Dennis is the patient here, it was the most successful one, but for other patients it also worked quite well, okay? So, and every proper activation of the exoskeleton sends, again, the proprioceptive signals because of the muscles and the stretching of the muscles and things like that, sends the proprioceptive signals back to the brain and helps the brain to recover, okay? So those are the ROC curves that we observed. Okay, this is false positive rate, this is true positive rates. They're pretty good. And if you compare it to the state-of-the-art research that has been published in healthy subjects, then you can see that what we see in the patient is not worse, or sometimes even better for those average ROC curves, which we see sometimes even better than those recorded from the healthy individuals. Okay, so that was the non-invasive part, and if I have time, I can talk 10 minutes about the invasive part of the interfaces. Peter, do I have time?
Yeah, well, I guess you still have a little bit of time. We started a little bit later, so yeah, feel free to.
Okay. So we under the funding obtained by Mikhail Lebedev from the Russian government, we started the Center for Bioelectric Interfaces, which meant to develop, the project was meant to develop the invasive brain-computer interface, something that Mikhail will be talking in more details. But I will show you like some development that we have made as well. So we made a partnership with University Clinic of Moscow State University of Medicine and Dentistry, together with academician Vladimir Krylov and the Dr. Mikhail Sinkin, who gets access to the patients who have the ECOG, electrocorticographic grids, implanted into the motor cortices for the reasons, for the purely medical reasons, for localization of the eloquent cortex, so cortical regions which cannot be removed during the surgery, or for localizing the seizure onset zones. Okay, we developed this setup where we're able to register activity of the hand, movement of the hand. We have the EEG device that record this electrocorticogram, and we are able to do some interesting things with it. The main goal of the project is to create a bidirectional brain-computer interface. So we're not only controlling the external device by using the activity, presumably, of the motor cortex, but we are also sending the information from the external prosthetic device onto the somatosensory cortex in order to add a tactile sensitivity or feedback, and then this was shown to improve the agency, and the person then thinks that the hand is actually his or her. Okay, so the first development was that we have created the online interface that is online, is able to decode the finger movements, so that here we can see that the person is moving her finger, and the finger, the avatar is moving as well. When she's not moving, the finger is not moving. Well, sometimes still moves, but of course there are errors. When she starts to slow down, when she starts to do it slowly, we will see it now. The hand is moving slower, okay? So that's the first part of the project where we basically implemented the control. And now we are in the process of the coronavirus, of course, slowed us a little bit, but now we are in the process of developing the stimulation of the cortex so that the person can move his hand and feel the result of this movement for when he or she touches something, okay? And we're thinking about some virtual movements of the hand that is touching like some I don't know, some Christmas tree toy, right? And if he or she touches too strongly, then the toy gets destroyed. But the only information about strong or not strong, he or she receives through the stimulation of the cortex. So, but during this project, we have also developed very interesting collaboration with the doctors and creating these interfaces, and building the decoders and looking at which electrodes. These grids show the electrodes. We have a grid of 64 electrodes here, 8 by 8. And the colors show that which of the electrodes appear to be utilized for decoding, okay? And that means that although the electrodes are overlaying the cortex in the locations where the specific function is represented, like in this kind, this is right, different fingers are represented, okay? And this way we can create a map for the neurosurgeon so that he or she knows that these regions are not to be touched, okay? So we tried different approaches with simple decoders, with more complex decoders, and it turns out that the complex decoders, like based on the convolutional neural networks, allow us to obtain more focal maps and therefore higher accuracy in localizing the eloquent cortex, which I think is very important when guiding the surgery, especially in the complicated cases when there is always a trade-off which cortical region you want to remove because of the epilepsy or because of the other, like oncology or other kind of tumors that happen, okay? And an example how you can map the sensorimotor cortex without stimulation, because like the standard procedure would be to open the skull, have a patient, look at the patient's hand, for instance, stimulate patient, okay? Apply electric current just directly to the cortex, okay? This procedure is very dangerous, especially in patients who have epilepsy, because this can cause a seizure. And that usually changes the operation, the flow of operation, and basically delays everything and endangers the patient quite a bit. Here is an example. The color is the red color you see, and this is the cortex that we're looking for. Here we were looking for the motor cortex, and indeed we can see we localize the motor cortex for the index finger in this case. And here there is a sensory motor cortex, sensory cortex localized for the same finger. Okay. So also we can do speech mapping, and also in a passive way without stimulating and looking, because the standard procedure is that the patient is woken up during the surgery and you show to the patient certain pictures of where there are certain actions or objects, and the person has to name, the patient has to name the objects or actions, and you stimulate and you see at which moment when you're stimulating which area this language function gets disrupted. Okay, so here we can do it absolutely passively. We just simply ask the person to name the objects, and then we analyze the activity and see where this activity changes, and then we can pinpoint the cortical region where the speech is represented, and then this is a taboo to touch by a neurosurgeon. Okay, we have gotten so far good agreement. We have now, tomorrow basically we have already the 5th patient now. This is an outdated slide. So where we will try to verify this technique. And hopefully soon it will become a part of the big complex for neurosurgical evaluation of the patients. Okay, so now I want to conclude. So basically, our colleague, late colleague, that made the study, Alexander Frolov, he made the study where he showed that the BCI actually improves the rehabilitation in patients with post-stroke patients. He did it for the upper limbs. We implemented foot motor imagery based BCI control over exoathletes, low impact the skeleton, and we are about to start clinical trials with this device. We are also actively working in the realm of invasive interfaces, because we believe that reading activity from the brain invasively with properly designed electrodes is actually the future of the BCI technology. And also what's very interesting, what's very encouraging is that developing this future application, we are already helping the patients and the doctors to handle the complex patient cases where we can provide the ways to monitor their brain activity and to localize cortical regions that should not be touched during the surgery. Yeah. So again, I want to thank all of you for listening to me, and that's the acknowledgement for the mega grant obtained by Mikhail Lebedev, and that stands for the Russian Ministry of Education. So heraldic sign. Yeah, thank you very much.